I don't know my credit card is.
I gotta put my camera up here.
I need to use good notes. Okay, I use both. I like Notability. And so like I worked with like a lot, and so he used Notability. So it's just like easier to like. Sure. Yeah. Sure. I was just gonna say I have. Helms. Let me share. Yeah. I was gonna make flashcards and stuff.
All righty. Good morning, everybody. How are we all doing? It's Friday, first week. We're supposed to be excited at the end of the first week, getting just pumped for the second week, right? There's only 14 weeks to go. Well, 15 if you count finals week.
15.
Okay. Any questions before we get started this morning? Which surgery group scheduled with me for 10:30 on Tuesday? I actually do have a student appointment that's in the middle of that time. It's not a problem. We're still going to plan on it, but I'm going to have to step away for a short period of time. But we'll get you underway if that has to happen. And it's a meeting I can't change. So that's the problem when I think without my calendar in front of me. So okay, and you guys want to go Tuesday at 6:15? Is that what you said? Okay, I just need to get on the calendar. I couldn't find your email, so your second email. So that's okay. I will try real hard to remember to just stick that on my calendar after class. Yes.
Should we be bringing a specific lab notebook to lab days?
So it all depends on how your group wants to keep lab notes. If you want to do them handwritten, that's fine. If you want to do them digitally, that's fine as well. But you are going to want to keep notes, and that's actually a good question, because you can never write down too much when you're doing an experiment. You can always not use information you write down, but it's really hard to use information that you didn't write down. So I would be documenting like crazy for these things. Case in point, when you're doing a surgery and something unique happens, unexpected happens, okay, then you document that. And then later on, when you're doing some analysis and you end up with an odd value for some reason in one of your experiments, and it turns out that it's related to that odd thing that's happened, then you have documentation of that, and it makes it a lot easier to kind of fold that into your analysis. Does that make sense? Yeah.
Do we need to bring lab coats, or do we need lab coats at all?
I would bring lab coats. Well, I mean, for the surgery, yeah, I would bring lab coats. I would, you're going to be wearing gloves during the surgery. And I would bring safety glasses too. Okay. There's nothing that we're going to be using, particularly for the rats, that's that's going to cause you to grow an extra limb or anything like that. But I just think it's good practice. And then when we're doing the rabbit surgeries, you really do want to have some protection for your clothes. And by the way, I would never wear anything that you would never want to get a stain on to our lab. When we do the rabbit surgeries, you're going to be cannulating a carotid artery. As you all know or will know, arteries are under high pressure. And if you nick an artery, it squirts, right? It's more scary than dangerous unless it's a really, really big hole. But you know, we've had multiple, usually there's a student every year who gets what we call the hematological baptism. And where you're just standing in front of one of those little pinpricks and it just squirts at you and you get a red droppy face. And probably good to have safety glasses on when that happens so it doesn't get in your eye or anything like that. Yeah.
How about like a suture and like scalpel kit? Do we need to bring our own?
No. I got all the stuff that you need. Anything else? Yes, Darian.
Sorry, just going back to the coats, will you have extras just in case? Because I definitely left mine.
Yeah. Yeah, I mean, we can get lab coats. That's not a problem.
Okay.
Stockroom has a ton of them. So as you could tell when we repaired all their wounds yesterday. Anything else? All right. Shall we get back to some content here? Okay. I want to share screen. I think that's what I'm gonna do. Where we go there. Don't mind me, I'm just talking to myself. No, that's the wrong one. I want that one. Actually, we'll start there. Okay. So this is where we left off last time. We were talking about adaptation, acclimatization, and acclimation. And we ended by talking about the fact that demonstrating that some physiological function is adaptive in nature is a really hard thing to do. And that the typical way that we study a system in order to determine whether or not it's adaptive is we will look at distantly related animals that live in very similar environments. And as we pointed out, they should evolve similar sorts of physiological mechanisms in order to tolerate that environment or function well in that environment. Alternatively, we can look at very closely related animals that live in quite different environments, and the idea being that if their physiology is adaptive, it should have diverged as they became optimally designed to work in these different environments, okay? And so we're going to look at an example of this. And this is an example you may have seen before. It's a pretty common example that shows up in a lot of Gen Bio texts. Oh, hang on. It's telling me to lock my iPhone. I don't know why. Which one? Oh, come on. No, I do. Okay, clearly pushed the wrong button there. All right, we got to wait a second. Now they're probably going to try to call me and ask me what the heck I need 911 for.
I don't think I actually called because it won't let you hang up with the calls. That's not really.
Maybe I need to get rid of it here. Oh, yeah, that would have worked. That would have saved me a lot of grief, wouldn't it? Look at me. No, we don't need the weather app. I need for my phone to wake up. All right, hang on. Stay tuned.
It's cold.
Oh, not again. Cancel. I've never had to go through this castle before. Finally, yes. All right, let's get back to business. All right, I think this is where we were. All right, what did I do with my little doodad? Technology. All right, two closely related organisms. The llama, the camel. Llamas live at high elevation in the Andes Mountains. Camels live in the Middle East, low elevation. Okay. The difference between these two environments is that when you are at high elevation, you are experiencing low oxygen levels. You get to high elevation, the density of oxygen molecules declines, therefore the partial pressure. And we'll talk about that when we get to the respiratory system. The amount of oxygen available for you to use is going to be less than it would be at low elevation. Okay, so this is a good example of closely related species that where we can compare their physiological performance based on the environments that they live in. Now, for years, it was thought that the fact that llamas were shown to have a type of hemoglobin that had a really high affinity for oxygen. So they could just latch on to this low density oxygen and use it in respiration. And they thought that this was an adaptation to them living at high elevation. But then they went and looked at hemoglobin in camels. And what did they find? The hemoglobin of camels also has a really high affinity for oxygen, even though they live at low elevation where that high affinity is totally unnecessary. What does this mean? That was a question. You are really smart students because you're just so well trained. Give me an answer. What does this mean? Yes.
It's likely not adaptive from their environment.
That's correct. It's likely not adaptive. So why do both of them have this characteristic? Oh, yeah. There you go.
Just waiting for a high five.
Yeah, I should have high five. Yeah, I should have done that. What is it? So what does that mean? Where does this trait come from? Go ahead. Yeah, it comes from a common ancestor. What it means is the high oxygen affinity of the hemoglobin is a taxonomic characteristic that's not been lost. in the camels. And the thing of it is if we understand how natural selection works, natural selection doesn't willy-nilly get rid of traits just because they're not needed anymore. Natural selection is driven by fitness. If having high oxygen affinity does not impact the organism's fitness, it's not going anywhere. Okay, so even though they don't need it and probably are even disadvantaged some way, and we'll talk more about that when we get to the respiratory system down the road, it doesn't really impact their fitness, so it still exists because they inherited it from a common ancestor. Key point to take away from this, when we're talking about physiological mechanisms and the adaptive significance of a physiological mechanisms, they're two completely different concepts. So we got to be cautious when we're trying to attribute some physiological characteristic to being a specific adaptation. Does that make sense? See one sort of head up and down head shake back there. Does that make sense? Okay, the next central theme, homeostasis. There isn't an organism alive that doesn't depend on some level of homeostasis. Homeostasis was first described by Claude Bernard back in the 1800s, when he was one of the first to notice that organisms worked hard to maintain physiological characteristics within some very, very narrow limits. This allows organisms to exist in environments that may fluctuate all over the place. This allows them to survive in these fluctuating environments, perhaps even stressful. Notice I put question marks after the word stressful, so I'm going to diverge here for a moment. It's really important that we use the term stressful cautiously when we talk about an extreme environment that anybody would live in. So if I were to ask you, what is the definition of physiological stress? How might you define it? Somebody take a crack at it. What's the worst that can happen? You get it wrong. That's okay. I get things wrong all the time. Yeah.
Some sort of physiological trait away from a set point.
Okay. It pushes a physiological trait away from a set point. So does that mean that organisms never exist successfully when a physiological trait gets pushed off a set point? We will talk a little bit later this morning how that actually happens all the time. But we're walking down the right road here. Anybody else? Let me rephrase the question. If you were going to look at any sort of organism, and we can even consider humans here. Humans are an organism. If you were going to look at them and evaluate them, how might you tell that whether or not they are physiologically stressed? Let me ask the question another way. Trying to get you to think, and I know it's early in the morning, but we can warm those brains up. If we were to go into the middle of the Saharan desert, the peak of summer, and we see an organism out there, would you consider it to be stressed? Yeah, go ahead.
I would say to like our norms, yes, but we don't know where the organism came from, because that could be its normal, environment, so.
Okay, that also contributes to what many would consider to be the correct answer. But what kind of indications might tell you that an organism is actually being stressed? Go ahead.
Placed under pressure.
Define what you mean by being placed under pressure. That sounds like student stress to me.
Like. Like put under conditions that aren't. In favor of the organism.
So how would you determine whether or not it was in favor of the organism? Go ahead.
You could measure things like blood pressure in the country. See if those very high, you can want only.
So let me ask you, what determines whether or not an organism is successful in an environment? From a biological perspective, what determines whether or not an organism is successful? Yeah, go ahead.
I'm just gonna say fitness, probably.
Fitness. What constitutes fitness?
Reproductive, is it efficiency or like health?
Reproduction,
Survival.
survivorship. Reproduction and survivorship. You should know that one. Okay. Reproduction and survivorship. So if the measure of success in an environment is judged by fitness, reproduction and survivorship, what should you look at to determine whether or not an organism is under stress? Yeah.
If it's dying and not reproducing.
If it's dying and not reproducing. Or, actually the key one there is probably reproduction. I think most would consider that an organism is existing in stressful conditions when they are unable to reproduce. They are investing so much in just pure survivorship. If particularly over the long term, they are unable to reproduce, then one might consider that that's under stress because that's impacting success. Does that make sense? So we've got to be careful about throwing that word stress around because an organism that is adapted to be successful in an extreme environment is not necessarily under stress. They're doing what they're designed to do. An organism that exists within its normal physiological tolerances, which means it probably is going to have the capacity to reproduce and survive, is likely not under stress. It's just tolerant of the extreme. Yeah,
So
just
you're saying, just want to double check that, the process of adaptation is not physiological stress. So if you like took an organism that lives in a hot environment, put in a cold, it also has to adapt.
actually what I would say that a little differently. What I would say is the components of the population which are under stress likely aren't going to survive. Those that have the genotypic and phenotypic ability, actually I will say phenotypic ability because that's what natural selection acts on, have the phenotypic ability to be successful in an environment will survive. So those that are under stress will not be passing genes on to the next generation.
So it goes.
And that's what will drive the adaptation. Does that make sense? Anything else? Wasn't that a fun discussion? All right, so homeostasis, getting back to homeostasis. Environments are widely fluctuating, and this really plays into what we've just been talking about. And organisms that are well crafted to exist in their environments have the capacity to maintain a good level of internal homeostasis in spite of what the external environment is doing. Okay. And this is really important. And all living things do this. Plants, animals, whatever. They all do this to some degree. And I will say that homeostasis can be maintained in a whole bunch of different ways. It can be physiological. It can be behavioral. Doesn't matter how it gets done, but the fact that it does get done is what is important. Let's look at an example here. I always like examples of roly-polies. Who hasn't played with a roly-poly, right? Okay. So there's a little isopod here. They eat decaying leaf litter, which is why you find them where you find them. They need help digesting that, just like anything that consumes plant material. And so they have bacteria that live symbiotically in their gut that require a pH of between 5.5 and 6 in order to work. And so Zimmer and colleague did a study where they were looking at how these roly-polies handle variable environmental pHs in the food that they're eating. And so what they did is they fed these guys food that ranged in pH from 2 to 8. And look what happens. Once they consume it, as soon as it hits the gut, that pH is adjusted. That pH is adjusted to right around 6 to make the gut environment suitable for those symbiotic bacteria that the roly-polies absolutely depend on. This is an example of gut homeostasis. Now, getting to your point about maintaining, how tightly do we have to maintain that level of homeostasis? Homeostasis does not mean that physiological parameters never vary, because they vary all the time. Over the short term, you can get substantial variation in physiological parameters. This graph here is a classic example of it. This graph plots blood glucose as a function of time of day. And look what happens each time we eat a meal. This is human data. We eat breakfast, we get a bump. We eat lunch, we get a bump. We eat dinner, we get a big bump. This guy kept making his meals bigger and bigger as the day got over. Okay. But even though we get these substantial bumps in blood glucose, over the long term, it's maintained around that set point. So when it deviates from the set point, we get a correction so that it comes back to the set point. This is what we refer to as dynamic constancy. So just because homeostasis is important, it doesn't mean that our physiology never varies from that homeostatic point. Because there are things that we do. You just need to have the capacity to get it back there so that it stays around that set point for the long term. Does that make sense? All right, here's another example. What happens when you get cold? So here's a flow chart right here. We're sitting in a room here. That's certainly not the case with this room. But we're in a room and the temperature gets cold. It will be the case with this room before this semester is over, though, I promise you. Because in this building, the heat works great during the summer. And I got to tell you, the air conditioning is amazing during the winter. So there you go. So room temperature declines. you start to lose body heat, because what happens is as the room temperature declines, the environmental temperature declines, the difference in temperature between the room and you gets bigger. We call that a thermal gradient. And the larger that thermal gradient is, the faster you passively lose heat to the environment, which means you get cold. When heat loss increases, your body temperature is going to decrease. And once your body temperature starts to decrease away from that set point, there are multiple things that occur. Your body's going to do a couple of things to try to reduce that passive heat loss. Okay, remember vasoconstriction? We've talked about that a couple of times. You are going to constrict, okay, the arteries that are serving the surface of your body, so that you can reduce blood flow to the surface of the body and reduce exposure of your core body heat to the surface of the body, which is where you lose it. And so that will reduce your heat loss. Another thing that you will do, and this is a behavioral response, is you might curl up. What does curling up do? It reduces your surface to volume ratio. It's exactly what it does. It makes your volume big and your surface area small, which means heat loss encounters a bottleneck. So that will also reduce heat loss. Then you'll up heat production. You might shiver. Do you know why shivering heats you up? This is a harder question. Yeah.
Because muscles make heat when they work, and so if you're working a bunch of muscles.
Do you know why muscles make heat when they work?
Because they're not perfectly efficient.
That's the right answer. That's exactly right. Muscles have actually a really low mechanical efficiency. You spend a lot of energy powering your muscles, but a huge proportion of that, probably under 20%, actually gets used to do mechanical work. The rest of it just gets converted to heat. That's why you sweat so much when you exercise. And so it's a good heat producer, and it's a great way to elevate your body temperature. And so this is a means of increasing heat production with the overall goal of returning your body to its set point temperature. All right. This leads us logically to the next central theme, which is intimately related to homeostasis, and that is feedback control. Feedback control makes the world go round. And these are regulatory mechanisms that are specifically designed in most cases, because I will show you an exception to this in a moment, in most cases to maintain homeostasis. This is why it's a key concept. And as we've already mentioned, organisms live in environments that vary a lot, constant state of flux. So the organism must constantly sample what's going on in its environment and then make corrections for that sampling. Key point right here. The degree to which an organism can tolerate an environment, okay, and this gets back to our discussion of stress. The key to which an organism can tolerate an environment is dependent on how big a correction it can make to an environmental perturbation. If I go and throw you out in the snow, your body will try to heat itself back up to compensate for the increased heat loss. But if that thermal gradient becomes so large that you cannot compensate, you can no longer elevate your metabolic rate to the point where you can compensate for that heat loss, then you will go hypothermic, because it's beyond your ability to tolerate. It's beyond your ability to make the correction. So this is just a generalized feedback mechanism right here. You are going to be sampling the environment, which is what the input means right here. And this will be put into some sort of a sensory system that's going to involve your central and peripheral nervous system. Your sensory system will ascertain what's going on and then construct an appropriate output for it, which will adjust what this input is going to be. And that's a simplistic look at what feedback does. Now, let's look at some specific types of feedback. We're actually going to start with a type of feedback that is not designed to maintain the normal state of homeostasis, but actually to deviate from it. And we call this positive feedback. Positive feedback mechanisms do some very important things. What they do is they will take a perturbation from a set point value, and amplify it, actually make it bigger. And there are some good reasons to do this. I'll give you an example in a moment. But a key thing to remember about positive feedback systems is that they are generally short-term in nature. They are designed to accomplish a specific task, and when that task is completed, you're going to revert back to your normal homeostatic state. It only takes a small disturbance under the right circumstances to initiate a positive feedback response. So the way that this works is you got a system that's being controlled in a homeostatic state. Okay, it's going to output a value. This value is going to be sensed by your sensor, your central nervous system here, and it's going to initiate some sort of a response. In this case, the response is going to be amplification. So it's going to feed back and increase the deviation from set point in this controlled system. Does that make sense? Here's just a graphical way to look at it. You got a set point here, perturbation from the value here, and then an amplification to what really would amount to a new short-term set point value. Okay, let's look at an example. The hormone oxytocin. Okay, oxytocin is controlled by positive feedback. And the reason being is oxytocin is one of the factors that drives uterine contractions during childbirth. So when proper stimulation occurs, the other thing that it does is it stimulates milk ejection from mammary glands. Okay, so when childbirth is occurring and you're experiencing stretching of the cervix, or you have a child suckling on nipples, it will cause an increase in the release of oxytocin, and that will compound over time. The last thing that you would want to do is to diminish uterine contractions during childbirth. That wouldn't work very well. And so here you have a positive feedback response that is designed to upregulate a system in order to accomplish a specific task. But once childbirth is over, then oxytocin levels will decline back to their normal set point value. Now, can you guys think of any other potential examples that would involve positive feedback? Go ahead.
Fight or flight response, is that positive feedback?
It could be, yeah. I mean, you're regulating actually a lot of things, and not the least of which is thyroxine levels. But it also is going to upregulate nerve response. which is tightly tied to a fairly classic positive feedback mechanism. Yeah, go ahead.
Would blood clotting be like an example of positive feedback system?
It could, yeah. When you are regulating things that are involved in clot formation, sure. Anything else? What about fever? Increase in body temperature. That is driven by a positive feedback mechanism. The most classic one that you've all heard of, nerve function. Without positive feedback, your nerves won't work. Because remember, when you have an action potential, what happens? You get that massive rush of sodium across the cell membrane that depolarizes the cell. That is absolutely positive feedback. So again, but in all these cases, we're looking at short-term needs that require the upregulation of the system. And that's the value of positive feedback. All right, that doesn't help us with homeostasis. But the important type of feedback with regard to homeostasis is what we call negative feedback. And in essence, negative feedback involves sensing the output of a controlled system and then correcting the response in the opposite direction of the deviation from set point. So if you get an increase from set point, then the response to that, so we have this inverting amplifier here, is going to be a decrease in the physiological response back towards set point. You're always moving the response back towards that set point value. Does that make sense? Okay. This is what it might look like here. So we get a disturbance, we make a correction. And in this case, the correction is an elevation above set point. The disturbance, I should say, is an elevation above set point. The correction is back towards set point. Note, we actually overshoot set point. And so we make another correction back towards set point. And we continue to do this all the time, in fact, to keep a physiological variable around set point value. Now, it's important to understand that a physiological parameter never flatlines on set point. You're always oscillating. We call these dampened oscillations around the set point value. Feedback mechanisms are not solely a property of physiological systems. You know, electrical systems use them all the time. Well, not this building's air conditioning, but you know, if you were to go to and run my my environmental chamber in my lab, which maintains an internal temperature that's relatively constant, you will see that it looks like a sine wave because it's constantly correcting the temperature to stay around the desired set point. That's the way feedback systems work. So if you were to measure it very precisely over time, you would see these dampened oscillations around the set point value. This is just another simple flow chart to kind of illustrate how it works. We've already talked about most of this stuff here. It just shows you've got some sort of a stimulus, which represents the output from that controlled system. It's going to be picked up by a receptor. Okay, this would be in the peripheral nervous system. That will then be delivered to the central nervous system, where it will figure out what to go do with itself. And note, it's going in via an afferent pathway. Anytime you see that term afferent, it tells you you're sending information to the central nervous system. The response will come via an efferent pathway, which is taking information from the CNS out into some sort of a controlled system. It will cause a change in that controlled system, which will be your response. And then the process will start all over again. All right, it's an example of negative feedback. Believe me, there are a whole host of them. This is just one. So let's stick with the whole concept of reproduction right here and look at the hormone prolactin. It is controlled by negative feedback. Prolactin, what it does, where oxytocin stimulates milk ejection from the mammary glands, prolactin actually stimulates milk production amongst a whole host of other things, and some of those are listed over here. All right. And so again, suckling is going to promote increased release of prolactin, which will increase milk production. Okay. But once it gets to too high a level, prolactin then will feed back negatively on the hypothalamus to trigger the release of dopamine. And dopamine shuts off prolactin production. So in this particular case, prolactin is regulating itself to some sense because it will actually promote this dopamine release, which will then start to close down prolactin production. So then prolactin will stay at a fairly constant value in circulation. Questions at this point? Now, some systems actually have multiple feedback mechanisms that are involved in its control. And the arrangement of feedback mechanisms that will control a particular system can actually be quite complex. So here's an example of the control of the release of thyroid hormone, which you guys will all become intimately familiar with. So the hypothalamus releases TRH, which is thyroid releasing hormone. This releasing hormone will then interact with the anterior pituitary to release thyroid stimulating hormone, which will then interact with the thyroid to release thyroid hormone, which comes in two flavors. I'm not going to tell you what these two flavors do, but you will figure it out. T3 and T4. And these two versions of thyroid hormone are each involved in their own separate negative feedback reaction. T4 is going to feed back on the hypothalamus and shut down the production of TRH. T3 will feed back onto the anterior pituitary and shut down TSH. The big point here is that when you have multiple feedback mechanisms that are involved in controlling a system, it gives you diversity in the way that you can control that system. It's really helpful if a system needs to be influenced by multiple things. You can have separate feedback mechanisms which can influence the performance of a system. It's really useful. Okay. One of your required readings is going to involve yet another type of feedback, which we're not going to talk about here. I'm going to let you read about it. It's called a feed-forward system. And those are actually quite interesting and will really tell you just how well designed physiological systems are to anticipate what might be happening in the future. We got a couple of minutes left. All right. Our last central theme, conformity and regulation. So when any sort of a living thing encounters environmental change, it's going to respond in one of two ways. It's either going to allow its internal environment to change with the external environment, which we call conformity. Organisms that do this, we call them conformers, or it's going to actively regulate its internal environment, through the use of, chemical, physiological, behavioral mechanisms to regulate that internal environment in spite of what's going on in the external environment. We call this regulation. And the organisms that do this, we refer to them as regulators. And I'll make this my last slide today. So these are graphs which illustrate what it looks like to be a conformer or a regulator. So these blue dots right here, okay, and this is a plot of the internal environment on the y-axis and the external environment on the x-axis here. The blue dots represent what we call the line of conformity. So if you are getting precise confirmation between the internal environment and the external environment, it would follow this line right here. The green line represents what the organism is actually doing. And what I want you to note here is that the green line does not extend the full length of that line of conformity. What does that tell you? Yeah.
They can't conform in like super extreme.
Okay, well, they can't tolerate super extreme. It's very possible that the line of line that represents the organismal response here is bound by lethal temperatures. So what this tells you is they are conformers, but conformers have to pick and choose where they live very carefully. This here is a regulator. What I want you to note about the regulator is there is a range of temperature over which regulation works really well, or there's a range of values, I should say. It could be temperature, where it works very well, but look what happens when we get outside the extremes. They become conformers. And so what this means is that even regulators have limits. Remember, we talked about the size of a correction that an organism make determines what kind of an environment or whether or not they're going to be successful in their environment when the environment is experiencing extremes. Well, when I throw you out in the snow, and you're regulating body temperature, I push you beyond this lower limit and you become a conformer and go hypothermic at that point. So even regulators are only regulators within the range of levels of correction that they are capable of making to regulate that value. Does that make sense? All right. We'll call it a day. Everybody have a great weekend.